Good afternoon, everyone. Today I'll be presenting on the immunology and the genetics of rheumatoid arthritis, which is a chronic autoimmune disease. We'll cover the genetic and the immune factors that contribute to the disease, the mechanisms behind how it manifests, and the current and emerging immunological treatments aimed at managing it. I'll explain both the roles of genetics and the immune system in RA and how they contribute to disease progression. And we'll also explore how therapies are improving the quality of life for patients. So let's dive in. So here's a very brief introduction of rheumatoid arthritis. So rheumatoid arthritis, or RA, is a chronic autoimmune disorder where the body's immune system mistakenly targets the synovium, which is the tissue lining the joints. So in this case, I'm talking about this section of the bone right here, which is the synovium. In the US, approximately 1.3 million adults are affected by RA, and it predominantly affects women, with female sex hormones being a contributing factor. RA typically develops between the ages of 30 and 60, though it can occur at any age. The causes of RA are multifactorial and not fully understood, but has a very strong genetic component, with 60% of cases showing a familial association. There's also external triggers such as smoking, hormonal changes, and infections, which also play a big role in the onset of RA. Understanding how RA develops is very crucial to developing targeted treatments, and that's what we'll explore today. And in this picture, I just want to highlight, this is just showing the difference between a normal patient with healthy joints, and this is a patient with rheumatoid arthritis, and it's highlighted with red. You can see that there's a lot of inflammation in the joints, which causes the deformity of the joints, which is a very common symptom seen in RA. So this is a very brief overview of the immune system's role in RA. I will be going into more detail on this subjects later on in the presentation. So the immune system plays a major role in causing RA, as I mentioned earlier. In people with RA, immune cells like T cells and B cells, they become overactive and they attack the joints. So these cells release pro-inflammatory cytokines such as tumor necrosis factor alpha, otherwise known as TNF alpha, interleukin 1, interleukin 6, also known as IL-1 and IL-6, which cause inflammation, worsening the damage to the cartilage and the bones in the joints. So another issue that is encountered in RA is that the body also produces autoantibodies such as rheumatoid factor, also known as RF, and anti-CPP2, which is known as anti-cyclic citrullinated peptide 2, which plays a major role in attacking the synovial tissue. So moving on, as I mentioned earlier, genetics play a crucial role in the development of RA. So studies have shown that about 60% of RA cases are inherited, meaning family history is a major risk factor. So a key genetic factor associated with RA is the DR beta 1 gene, which is located on chromosome 6. So this gene is part of the MHC class 2 complex, which helps the immune system distinguish between self and non-self. So the DR beta 1 gene is responsible for about 30% of the genetic risk for RA. Twin studies have been instrumental in showing the genetic influence on RA. For example, if one identical twin develops RA, there is a 12 to 15% chance that the other twin will also develop it. However, this risk does drop to just 4% in non-identical twins, which highlights the importance of genetic predisposition. So the genetic factor is not the sole cause of RA, but it significantly increases the likelihood of its development. So moving on, we will discuss the shared epitope hypothesis, which offers an explanation for the genetic predisposition in RA. So this hypothesis suggests that certain variations in the DR beta 1 gene increases the susceptibility to the disease. So the MHC class 2 molecules, particularly the DR beta chain, are responsible for presenting antigens to the immune system. In individuals with RA, specific amino acid sequences in the DR beta 1 chain are shared across multiple RA susceptible alleles, and these sequences, termed the shared epitope, are thought to make the immune system more likely to attack the body's own tissues, specifically the joints. So, the most well studied allele associated with RA is in fact the DR beta 1 0401. And this hypothesis has been pivotal in understanding how genetic factors influence immune responses and contribute to RA, helping to link specific gene variations to disease susceptibility. So, in order to fully understand the shared epitope hypothesis, it is crucial to understand the genetic organization of the class 2 region. So the MHC class 2 region contains at least 14 different genes, in which the genes are divided into 3 main subregions, which are the DP region, the DQ region, and the DR region. And each subregion, as you can see in the diagram, contains functional alpha and beta chains, which are known to be polymorphic. And these polymorphic chains are crucial for forming membrane-bound heterodimers. Moving on, of the 3 main subregions, which again I'm emphasizing are the DP, DQ, and DR, the shared epitope hypothesis focuses on the DR subregion, and specifically the DR beta of the DR subregion. And this picture displays the DR beta region. So this chain, which is the DR beta chain, contains regions of variability which are specific sections of the genome where the genetic variations are most likely to occur. So the notable region is the hypervariable region, which is this region pictured below. And this region consists of 3 regions. It consists of HV1, HV2, and HV3. The HV3, which is what the epitope hypothesis focuses on, is hypothesized to play a critical role in the shared epitope hypothesis, in that it is the region in which the sequence differences occur between the DR4 alleles, which again, it's one of the genetic variants of the DR gene, which determine the susceptibility of one acquiring an autoimmune condition, specifically in the context of RA. And as I mentioned previously, the DR4 molecule with the strongest link to RA is the DR beta 1 0401, which I mentioned on this slide over here, which I'm going to highlight. So in this upcoming slide about the immunology behind the RA manifestation, which on the next slide it goes into more detail, it talks about number 1, the citrullination of proteins which cause immune system confusion, and it talks about how activated T cells promote inflammation, and how interferon gamma and tumor necrosis factor alpha activate B cells, leading to autoantibody production. And so this is an in-depth section where I will talk about the RA pathogenesis. So at the early phases of RA, a process called citrullination takes place, where the amino acid arginine is converted into citrulline. However, due to many genetic dispositions as well as secondary external factors, APCs fail to recognize these proteins as self-antigens and carry these self-antigens to the lymph nodes, which initiates the activation of the CD4 plus T cell through an interaction with the TCR, which is the T cell receptor, and the MHC class 2 peptide antigen. Also, there are two classes of the CD4 T helper cells that reside in the synovial tissue, which is the CD4 T helper 1 and the CD4 plus T helper 17, which these two T helper cells can be differentiated by their distinct cytokine profile. The CD4 T helper 1 releases cytokines such as interferon gamma, tumor necrosis factor alpha, and lymphotoxin B, which all directly activate B cells, as you can see over here. These B cells then differentiate into autoantibody producing plasma cells that secrete antibodies such as RF, also known as rheumatoid factor, and it also secretes anti-CCP. After they are secreted, these antibodies then enter the blood circulation, as you can see here, to reach the joint. So due to the RF, which I'm going to go and zoom in a little bit. So due to the RF being an IgM antibody, it targets the Fc portion of the IgG, forming an immune complex that causes the inflammation of the joints. In the meantime, citrullinated peptide encounters anti-CCP, which activates the complement system, leading to this destruction of the joint cartilage specifically. Moving on, T effector cells, which is this cell over here, it stimulates the macrophages and the fibroblasts in the joint cartilage to secrete TNF alpha, which increases the adhesion to molecules in endothelial cells, which can be seen over here. The macrophage is what secretes the TNF alpha, which then promotes leukocyte migration into the joint. At the same time, macrophages also secrete inflammatory cytokines such as IL-1 and IL-6, which further advance synovial inflammation while also recruiting more inflammatory cells to the joints. These very cytokines contribute to the expression of rank ligand, which you can see over here. And this is the cytokines. I mean, it's kind of cut off, but there's interleukin 1, which is this one, and interleukin 6. And rank ligand, also known as rank L, it is expressed on the surface of the T cells, which bind to and activate pre-osteoclasts. And you can see this is the activation of the pre-osteoclasts. And after the pre-osteoclast is activated, it goes and becomes an it's from pre-osteoclast and it becomes an osteoclast. And the presence of osteoclasts, as we know by their nature, is what's responsible for the aggressive bone destruction in RA, which is shown in the later stages of RA. In the meantime, the inflammatory cytokines mentioned above also activate fibroblasts, which are synovial cells, and they secrete MMP, which contribute to cartilage degradation and destruction. So moving on, currently there is no cure for RA, but there are several therapies available to manage the symptoms and slow disease progression. First, NSAIDs, which are also known as non-steroidal anti-inflammatory drugs, are commonly used to relieve pain and reduce inflammation. These work by inhibiting enzymes like COX-1 and COX-2, which are involved in the production of pro-inflammatory molecules. Steroids such as prednisone help modulate the immune system and reduce inflammation. DMARDs, which are also known as disease-modifying anti-rheumatic drugs, are another key treatment class. So conventional DMARDs like methotrexate target general immune responses, while biological DMARDs such as TNF inhibitors target specific inflammatory cytokines like TNF alpha. And these therapies significantly improve the quality of life for patients, but there is still a need for more personalized treatment. Ongoing research into biological drugs and targeted therapies holds promise for even more effective treatments in the future. So, in conclusion, rheumatoid arthritis is a complex autoimmune disease with both the genetic and immune factors contributing to its development. The DR beta 1 gene is a significant genetic factor, while immune dysregulation involving autoantibodies and inflammatory cytokines plays a critical role in the disease process. While treatments like NSAIDs, steroids, and DMARDs are effective at managing symptoms, ongoing research into more targeted therapies offers hope for the future improvements in patient care. With better understanding and treatment options, we can expect to see advancements in RA management that significantly enhance the quality of life for patients. Thank you so much for your attention.